Okay, hi, I'm Jess Ward. I'm an associate professor at Ball State University. And so today I'm going to talk a little bit about the project that we were running, looking at the effects of microplastics on early life stage behaviors in fish. And before I get started, I'd like to mention that my co-author on this project is Kenzie Persinger, who did a lot of this work as a part of her master's project. Next slide. Microplastics are a growing environmental concern in a lot of freshwater and marine waterways. And so the main objective of our study was to evaluate the effects of microplastics on embryos and larvae, early life stage behaviors. And we did this in two ways. We're looking at the effects of microplastics alone on embryos and larvae of fish. And then we're also looking at the potential for microplastics to serve as vectors for other aquatic contaminants that are known to have adverse effects on aquatic wildlife. And so the contaminant that we were using in combination with microplastics in this case was 17-alpha ethanol estradiol, or EE2, which is the synthetic estrogen that is used in birth control pills. So this project had two experiments associated with it. First, we were looking at the effects of exposure to microplastics alone or in combination with EE2 on larvae after hatching. So we exposed them for 14 or 21 days after hatching, and then we looked at larval behaviors. In our second experiment, we looked at what the effects were if we exposed the adults, the parental generation, the F0 generation, and then looked at what the downstream effects were on embryos and larvae. And so we had 4 treatments associated with this project: a control treatment, a virgin microplastic treatment, and then microplastics associated with either a low or a high environmentally relevant concentration of EE2. So I can say that when we ran our first experiment, when we exposed larvae beginning 1 day after hatching for 14 or 21 days, we see weak trends towards hyperactivity in those larvae when we test them in behavioral trials. So we look at open field trials where we put them in arena and we have automated behavioral tracking software to record what their movements are. So we see weak trends in that respect. However, when we expose the FO generation and then we look at the downstream effects on the F1 generation, we see much greater results on embryos and larvae. So if we take parents, we expose them for 30 days, we collect their embryos, which is what you see in that first set of panels there. And then at 4 days post-fertilization, we assess their activity levels in the chorion using tracking software. And so when we do that, we see quite a bit of hyperactivity happening in these embryos, in those embryos whose parents had been exposed to microplastics either alone or in combination with the E2. Then we hatch out those embryos, we raise them in larval form for another 14, 21 days. Half of them will receive no further exposure, the other half receive continued exposure, and then we test them again. And in the larval stage, we also see trends towards hyperactivity in behavior in the larvae in open field trials. We also see changes in risk-taking behavior as well, which is what you can see in these heat maps over here. So larvae that have not been exposed to any kinds of microplastics, they're performing innate behaviors, they're staying close to the edges of the arena. They are not venturing into the center of the arena, whereas larvae that have been exposed to microplastics are venturing into the center of the arena more. This is a proxy for risk-taking behavior that has been shown to correlate with increased mortality in the field due to predation. So our next steps are, we're just finishing up this transgenerational experiment. We have one more treatment to test. And then our longer term goals, our next steps would be to determine whether or not these changes, these developmental changes in behavior that we see, hyperactivity in the lab, translate into actual changes in individual level fitness in the field. And then how can we use those data via modeling approaches to predict what these individual level consequences are for populations and communities downstream in natural environments. Thanks.